So I realized at a young age that heart disease was a serious issue in my family. And at the time, it seemed like something that I just had to accept.
I saw lots of members of my family suffering from atherosclerosis and heart disease. And so when I became a scientist, I saw it as a great opportunity to study diseases that are both a global problem, but also very personal to me.
So ApoB is the protein at the heart of heart disease. It's a massive protein that interacts with fats and lipids and forms low-density lipoprotein, or LDL, what most people call bad cholesterol.
It was so difficult to study because it has a really unique structure.
Being able to see the structure of a protein is like being able to visualize the shape of a Lego block. And that shape gives you an indication of where this particular protein might fit and what role it might play in the human body.
Getting a protein structure would take a year or more. It's a highly uncertain process. And AlphaFold lets them get an answer in 5 minutes instead, and that lets them get on to the next part of their work.
And that completely changes the way you conduct research. So it accelerates research, it democratizes research, and it expands what is possible.
When we finally saw ApoB 100 structure clearly for the first time, it was the first real eureka moment for me as a scientist, which is so exciting.
So AlphaFold has completely changed the rhythm of my lab's research. We use these structures as raw material for simulation-driven research that simply wasn't possible before.
Now we can have a very accurate prediction of what that looks like before ever going to the microscope.
This research into heart disease is such a lovely example of how you go all the way from kind of AI and atomic understanding of something out to whether we get more effective treatments for heart disease, whether we understand the progression of this, whether we understand the biological mechanisms involved.
We solved 200 million protein structures and gave it out to the scientist. So there will be much more breakthroughs with the faster space to advance science across from finding malaria drugs to helping crops to be resistant for the heat to plastic-eating enzymes.
The value of publishing this paper through Springer Nature gave us the key elements on what we should be prioritizing in terms of exposition, the types of questions that reviewers were interested in us focusing on, and really made the paper much better.
There's been a good relationship between Google DeepMind and Springer Nature over the years. The role of Springer Nature is to help ensure that the work that DeepMind is publishing with us is seen as being trusted, it's seen as being vetted, it's seen as being accessible.
On AlphaFold, one of the really big benefits we got were the peer reviews that our editor, Angela, facilitated. And in fact, one of the comments on a research question led us to do a bit of new research and build another piece, PAE, which became a quite important part of scientists understanding AlphaFold's confidence.
Open access is incredibly important. When AlphaFold 2 was published, researchers immediately seized upon the information in the paper, the database, the code, started exploring it, building upon it, and creating that real-life impact from the discovery.
So solving the structure of ApoB with just one piece in a much larger puzzle, but it was a crucial one that can potentially affect how we think about cardiovascular disease more broadly, and potentially improve the lives of millions of people.